There is a gesture that millions of people repeat every day without thinking too much about it. A glass of water, a
Drink plenty
capsule
of
or a
water.
small tablet,
Take your
a
medication.
swallowing
This diagram
movement,
illustrates the
and
mechanism of
then
swallowing and how
nothing.
the
Or
digestive
at
process
least,
begins
that
in the
is how it
esophagus.
seems from
Food
the
travels to the
outside.
stomach for the clock
What
marks external
occurs in the
time,
seconds,
while the body
minutes, and
synchronizes internal
hours
processes.
following
After
that moment
swallowing, the
is one
capsule
of the
enters
most
the esophagus.
extraordinary
The stomach's muscles
processes the
contract
human
in
body
waves,
can
churning
execute
food. The
without
autonomic nervous
anyone
system controls
consciously
body functions
ordering
without
it. It is a process that pharmacologists took centuries to begin to understand, which still holds surprises, and which transforms a molecule manufactured in a laboratory into a biological response capable of stopping an infection, calming pain, or stabilizing a heart beating out of rhythm. What is about to happen inside the body after swallowing that pill is not simple. It is a story of transport, transformation, molecular recognition, and, in the best-case scenario, healing. It is a story that no one tells you when the doctor hands you a prescription. For most of human history, the question of how a medicine worked had no answer, because no one knew it had to be asked. Remedies existed. Of course, willow bark relieved fever, opium calmed pain, and mercury was used against syphilis, often with devastating consequences. But no one understood why some substances did something while others did nothing and others directly killed. Medicine was then a mixture of empirical observation, tradition, and pure intuition. It was not until the 17th century, with the birth of modern chemistry, that scientists began to suspect that the effects of medicinal plants were not due to the entire plant, but to specific compounds within it. And it was not until well into the 19th century that someone was able to isolate one of those compounds for the first time. In 1804, the German pharmacist Friedrich Serturner extracted a pure substance from opium that he called morphine, in honor of Morpheus, the Greek god of sleep. It was the first time in history that an active principle of plant origin was isolated. The world did not know it yet, but modern pharmacology had just been born. From that moment, the question stopped being just, what cures, and began to be, how does it do it? And that question, which seems simple, has occupied thousands of scientists for more than 2 centuries. Let us return then to the moment you swallow that pill. The first thing that happens is mechanical. The tablet reaches the stomach, but the path there is no longer trivial. Some pharmaceutical forms are designed to begin dissolving in the mouth, taking advantage of saliva and the small absorption surface offered by the oral mucosa. Others are coated with materials that resist gastric acid, because the medicine they contain would be destroyed in the stomach before being able to do anything. These are called enteric tablets and represent one of the first tricks of pharmaceutical engineering, the art of delivering a molecule to the correct place within the body. In the stomach, dissolution continues. Hydrochloric acid and digestive enzymes begin to disintegrate the capsule or tablet. But the stomach is not the final destination for most drugs. It is rather a transit station, a place where the solid form becomes a solution. And from there, most of the real work begins a little further down. The small intestine is the great absorber of the human body. Its interior surface, if it could be completely extended, would cover something like the area of a tennis court. Millions of microscopic villi and microvilli increase that surface exponentially, creating a contact zone between the outer world, what we eat and drink, and the inner world, the bloodstream. It is here where most of the drugs we ingest pass from the digestive tract into the blood. This process is called absorption, and it is not simply that the drug molecule filters into the blood like water through a sponge. It is something more sophisticated. The cells lining the intestine have specific mechanisms. Some small, fat-soluble molecules can diffuse directly through the lipid bilayer of the cell membrane thanks to their affinity for fats. Others, larger or more polar, need to be actively transported by specialized proteins that act as molecular gates or shuttles. and others simply cannot cross this barrier well, which explains why many medications that work wonders when injected are useless when taken orally. Once absorbed, the drug molecule does not go directly to where it needs to go. There is one more obstacle, a filter that ancient medicine completely ignored and that modern pharmacologists consider one of the most determining factors in a medication's effect. It is called the hepatic first-pass effect. Everything absorbed in the intestine first reaches the liver through the portal vein. And the liver, that extraordinarily active organ that functions as a precision chemical factory, does not distinguish well between a nutrient and a drug. Its job is to metabolize, transform, and in many cases, inactivate substances. So, a significant fraction of the drug that has just been absorbed in the intestine is modified or destroyed before reaching the rest of the body. For some medications, this effect is so intense that only a small proportion of the original dose reaches the general bloodstream. Pharmacologists call this proportion oral bioavailability, and it is one of the first factors studied when developing a new drug. Nitroglycerin, used to relieve pain in angina pectoris, is a fascinating example. Taken orally, the liver destroys it almost completely before it reaches the heart. That is why cardiologists administer it under the tongue, where absorption is direct, rapid, and avoids that first hepatic pass. An elegant solution to a biological problem that for a long time was a mystery. Once the drug has overcome all these obstacles and enters the bloodstream, another equally complex phase begins: distribution. The blood carries the molecule to all tissues of the body, but not all tissues receive it equally. Some drugs dissolve easily in fatty tissues and accumulate there for hours or days. Others bind to plasma proteins like albumin, which function as a kind of bound transport. While the drug is bound to this protein, it cannot exert its effect because only the free fraction of the molecule is pharmacologically active. This free fraction is what actually reaches where it needs to go, and here is where the story becomes truly molecular. The human body works through signals, chemical signals between cells, between organs, between the nervous system, and the rest of the organism. These signals are transmitted through messenger molecules such as hormones, neurotransmitters, or cytokines that bind to specific receptors on the surface or inside cells. Imagine a lock and a key. The receptor is the lock, and the messenger molecule is the key that opens it, triggering a response inside the cell. Drugs, in their vast majority, work by interacting with these receptors, and they do so in two fundamental ways. Some act as agonists. They mimic the body's natural messenger molecules, bind to the receptor, and trigger the same or a similar response. Morphine, for example, binds to opioid receptors in the brain and spinal cord, the same receptors activated by the body's natural endorphins during exercise or pleasure. By doing so, it inhibits the transmission of pain signals and produces that powerful analgesic effect. Other drugs act as antagonists. They bind to the receptor but without activating it, physically blocking the entry of natural messenger molecules. Beta blockers, widely used in cardiology, block the beta-adrenergic receptors of the heart, preventing adrenaline from excessively accelerating the heart rate. The heart beats slower, with less force, and blood pressure drops. A molecular blockage that translates into years of life gained for people with hypertension or heart failure. Although receptors are the target of action for a large part of modern medications, not all act this way. Some inhibit enzymes, others block ion channels or transporter proteins, and many antibiotics directly interfere with essential structures of bacteria or other microorganisms. In pharmacology, there are numerous mechanisms of action, but all share the same goal: to modify a biological process to produce a therapeutic effect. But there is one more layer of complexity that makes the effect of a medication not the same for everyone, which for a long time was completely unknown. Receptors are not the same in all human beings. The hepatic enzymes that metabolize drugs also have genetic variants, or polymorphisms, that make some people degrade certain medications much faster than normal, needing higher doses to obtain the same effect, while others degrade them so slowly that a standard dose can accumulate to toxic levels. This reality, which pharmacology began to study rigorously in the second half of the 20th century, and which we know today as pharmacogenomics, explains many of the mysteries that for decades doctors could not resolve. Why did a drug work extraordinarily well in some patients and was completely ineffective or dangerous in others? Codeine is one of the most illustrative examples. Administered as an analgesic, codeine is actually a prodrug, an inactive molecule that the liver converts into morphine through an enzyme called CYP2D6. People who have extra copies of the gene that codes for this enzyme, called ultra-rapid metabolizers, convert codeine into morphine at such a high speed that they can reach potentially dangerous concentrations with doses that would be safe for most. In contrast, slow metabolizers, who have a poorly active version of that enzyme, barely convert codeine into morphine, and the analgesic simply has no effect on them. This is one of the reasons why personalized medicine, the idea that treatments should be adapted to the genetic profile of each patient, has gone from being a theoretical aspiration to becoming a growing clinical reality in recent years. Meanwhile, while the drug is doing its job, the body does not remain passive. It is already working to eliminate it. The elimination process occurs mainly in two places: the liver, which transforms the drug into more water-soluble metabolites to facilitate excretion, and the kidneys, which filter those metabolites from the bloodstream and expel them through urine. Some drugs are also eliminated through feces, breath, sweat, or breast milk, which has important clinical implications, especially in lactating women. The time it takes for the body to eliminate half of the amount of a drug in the organism is called the elimination half-life. It is one of the most important parameters in pharmacology because it determines how often a medication must be taken to maintain stable therapeutic concentrations in the blood. A drug with a half-life of 4 hours needs to be taken several times a day. One with a half-life of 24 hours can be taken once, and there are extreme cases. Fluoxetine, a widely used antidepressant, has such a long half-life that it can remain active in the body for weeks after stopping it, which profoundly influences how its start and withdrawal are managed. There is something that deserves to be said at this point because a persistent confusion surrounds medications in popular culture. Many people assume that a more potent medication or a higher dose necessarily produces a proportionally stronger effect. But the relationship between dose and effect is rarely linear. Pharmacologists represent it with what they call dose-response curves, which have a characteristic S shape. At very low doses, the effect is minimal. Then there is a zone where small increments produce large changes, and finally, the effect reaches a plateau where increasing the dose no longer produces more benefit, but only more risk of toxicity. It is the principle that Paracelsus, the 15th century Swiss alchemist and physician, expressed with a phrase that has survived 5 centuries. The dose makes the poison. Every substance can be toxic in sufficient quantity, and almost every substance can be useful in the right amount. This principle is the heart of toxicology, and also one of the most misunderstood ideas by the general public, who often perceive medications as good or bad in absolute terms, without understanding that their benefit or harm depends entirely on the context, the dose, the person, and the moment. The history of adverse effects is itself a story that deserves to be told because drugs are not magic bullets that hit the target with perfect precision. Selectivity, the ability of a medication to act only where it is intended to act and nowhere else, is one of the great challenges of modern pharmacology, and, in many ways, one of those furthest from being completely resolved. The receptors on which a drug acts are not always only in the diseased organ. The beta-adrenergic receptors that beta-blockers block in the heart are also in the bronchi. Therefore, in people with asthma, certain beta-blockers can trigger a dangerous bronchospasm. The same mechanism that saves the heart can compromise breathing. Non-steroidal anti-inflammatory drugs like ibuprofen inhibit enzymes called cyclooxygenases that produce pro-inflammatory prostaglandins, which relieves pain and inflammation. But those same prostaglandins also protect the gastric mucosa, so inhibiting them can cause ulcers or digestive bleeding in susceptible people. These are not avoidable imperfections. It is about the nature of biology. The body's systems are deeply interconnected, and a chemical intervention in one of them rarely leaves all others completely intact. The search for more selective, more precise drugs has driven decades of research and continues to do so. Rational drug design, based on detailed knowledge of the three-dimensional structure of receptors, has allowed the creation of molecules that fit their molecular targets with much greater precision than drugs discovered by chance or trial and error. Selective serotonin reuptake inhibitors, the antidepressants known as SSRIs, emerged precisely from that effort. Compared to tricyclic antidepressants that blocked multiple neurotransmitter systems at once, with a significant adverse effect profile, SSRIs act more specifically on serotonin, with better tolerability for most patients. And yet, even the most careful design does not always predict what will happen when a molecule encounters the complexity of a real human organism with its genetic variability, concomitant diseases, other medications, diet, and intestinal microbiome. That last element deserves a special mention because it represents one of the most active frontiers of current pharmacological research. The intestinal microbiome, the trillions of bacteria, fungi, and viruses that inhabit the human intestine, is not a passive passenger in the story of a medication. Evidence accumulated in recent years suggests that the microbiome can metabolize certain drugs, modify their chemical structure, and alter their effect in ways we are only beginning to map. Some studies indicate that the composition of the microbiome can influence the response to certain antidepressants, chemotherapies, and even modern immunotherapeutics used in cancer treatment. There is not yet a consensus on the real scope of these interactions, and research in this field is in full expansion, but the image emerging is clear. The effect of a medication depends not only on the molecule and the patient, but also on the millions of microorganisms that live with them. Pharmaceutical nanotechnology, for its part, is redefining another of the fundamental questions in the history of medicine: how to get exactly where you need to go. For decades, one of the great problems of oncological chemotherapy was precisely this. Chemotherapeutic molecules were deeply toxic to tumor cells, yes, but also to healthy cells that divided rapidly, such as those of the hair follicle, the intestinal mucosa, and the bone marrow. Administering a cellular poison and hoping it would preferentially accumulate in the tumor was for a long time the only option available. Targeted delivery systems, nanoparticles loaded with drugs designed to recognize specific markers on the surface of tumor cells and release their content directly into them, represent one of the most promising advances in pharmacology in recent decades. Some already have clinical approval. Many more are in clinical trials. The promise is immense, although the path from the laboratory to the patient remains long and full of obstacles that science is solving one by one. This entire journey, from the first contact of the tablet with saliva to the last molecule eliminated by the kidney, is what pharmacologists call pharmacokinetics, what the body does to the drug, and it is distinguished from pharmacodynamics, what the drug does to the body. Two complementary perspectives on the same process, two different lenses to observe the same story. There is something deeply human in the fact that for centuries people ingested substances that relieved them without understanding at all why they did so, that they trusted in the accumulated empirical knowledge of generations, in the patient observation of what worked and what did not, without having access to any of the conceptual tools we take for granted today. And yet, with that limited vision, they found remedies that in many cases turned out to be truly effective. Modern pharmacology, with all its molecular precision and its ability to design molecules to fit specific receptors like pieces of a biological puzzle, is actually the heir to thousands of years of that empirical observation. Every contemporary medication carries in its history traces of plants, of alchemists, of doctors who tried things without knowing exactly why they worked, of errors that ended up revealing unsuspected mechanisms. Aspirin was originally synthesized as a milder version of the salicylate from willow bark. Penicillin was discovered because Alexander Fleming forgot to clean a petri dish before going on vacation. Viagra reached the market as a treatment for pulmonary hypertension, and it was only during clinical trials, thanks to the fact that male volunteers did not want to return the medication at the end of the study, that it was discovered to have a side effect no one had anticipated. The history of medications is full of fortunate accidents, of paths that lead to unexpected destinations, of questions that generate answers, and answers that generate new questions, and behind every tablet there is exactly that. A process that does not happen all at once, but in stages. A story that begins in the intestine and is written in the bloodstream. A molecular dialogue between a substance designed in a laboratory and a body that has spent millions of years refining its own internal communication systems. The next time someone swallows a pill, the gesture may seem just as simple. But beneath that apparent simplicity occurs something that science took centuries to begin to understand and that still, in many aspects, continues to reveal new layers of complexity. Because biology is not simple, and the medicine that tries to speak to it is not either. What is certain is that every advance in the understanding of that journey, from the mouth to the cell, from the molecule to the patient, has represented a small but cumulative victory of human curiosity over disease, and that accumulation over generations is what has transformed diseases that a century ago were death sentences into manageable conditions, and has turned that daily gesture of swallowing a pill into one of the most scientifically charged acts that exist. If the journey of a simple pill through the human body reveals anything, it is that behind every medication there is a story much larger than the disease it treats. Centuries of curiosity, fortunate mistakes, precisely designed molecules, and organisms that never cease to surprise those who study them. If this story changed the way you look at the small tablets in your medicine cabinet, please subscribe and like the channel. We are uncovering the history they tried to bury. Let's plant the future. The next treasure will be opened soon.